Hello students, am I clearly audible and visible? Quickly let me know in the chat box and share the link with your friends, those who are thinking that the class is at 4 and classes at 5 and not at 4. Okay, quickly share the link with your friends and let me know in the chat box if I am audible and visible. Quickly. Yes, let's check. Yes, let's send the notification. Yes. Hello. Okay. So hello students, first of all, Happy New Year. We have entered 2024 and this is the first class of 2024 on Vedantu NEET English, and myself Gopika ma'am has come here on your first class. So I hope everyone celebrated New Year really well and all of you are wishing for so much more this year. And I also hope, as a teacher, I hope that this year most of you or all of you are in your dream colleges and getting good ranks in your NEET, because that is what this year is about. This year is very important to us as teachers and as students. For you people, we do not want to see you next year as NEET aspirants, but we want to see you people as MBBS first year students, second year students, right? That is a big dream. So I hope we will be able to do this dream, right? Okay. Yes, sure. Your syllabus will be maximum. We will try as much as possible to complete it, and whichever chapters you people are requesting us to do, we'll definitely take that first, because we are going on priority basis, because you already know. So depending on everything, we will be giving priority to the chapters that you want in real need. Okay, like there's no point in me taking biological classification if the entire crowd already knows the chapter, because the chapter is very simple and you have already started the preparation of this chapter, even it's a board student or even if it's a NEET student, right? But then at the same time, there would be a lot of students who want human health and diseases or biotechnology. That will be my priority. Clear? So happy new year to all of you. Let's hope this year brings a lot of happiness and joy to all of us and to our Vedantu NEET English, and let's grow as a family. Let's make it 200k very soon. So subscribe to our channel if you still haven't subscribed to our channel. Okay, so hello students, I am Gopika, your master teacher in Vedantu NEET English channel. I'll be taking biology or your zoology in specific. So today we are going to do HGP and DNA fingerprinting, which I did not do in molecular basis of inheritance because the concept was really wrong and the length of the video also was really long. So I thought, okay, let me take this as completely a one portion, because here there are a lot of tricky things which are not mentioned very clearly in NCERT, but definitely a teacher can break it down for you people. Okay, so I'm so happy to see you all. Thank you so much students, thank you. And if you still haven't watched our special Happy New Year video, go and watch, where all four of your teachers are there and we are telling about our college days, our school days, and how we prepared for NEET and so much more. Okay, yes. Students, botany will be handled by Baswaraj sir and it will be easy for you people. But if there is any concept that you need us to take in very urgent, like some cycles, like photosynthesis is done, respiration will be done this week. So is there some concepts that you find really difficult? Please let us know in the comments of this class. I will definitely take it up. Okay, yes. So students, if you want to join us on Vedantu NEET English Telegram channel, this is the link. Okay, you can join us. And also, if you are still not part of the Super 6 series, it's really sad because it's already New Year and your first resolution should be to join your teacher's life. Okay, students, let's start HGP. So HGP's full form is Human Genome Project. Okay, so in this whole molecular basis of inheritance, we have been speaking about very few things. Okay, all of you are with me? Did you all hit the like button? Why I cannot see the numbers increasing? Why is there so much conjuicy? Okay. Students, test results I have shared it on my Telegram as well as on Vedantu NEET English Telegram. Our mock test 2, right? That test results we'll be sharing this week. Okay, we'll be sharing this week. Do not fear. Thank you, Pandu. No fear when Gopika ma'am is here. Very nice. Okay, so students, in this chapter, molecular basis of inheritance, we have been talking only about few things. That is basically your DNA getting converted into mRNA, and that mRNA forming protein. this is what the whole chapter is about, correct? If you all look, this is your central dogma, and if you look at the central dogma, we will see replication, which we already discussed. We have done all of that very nicely, right? From DNA to mRNA is called transcription. Is there any student who does not know this? Then we have translation. So other than these processes, other than your transcription, translation, replication, semi-conservative replication, and understanding your nucleotides, what else is there in this chapter? That is where you come with the importance of HGP. Okay, students, so when I talk about HGP or your Human Genome Project, which most of you put it in the chat box, okay? See, the full form of HGP is Human Genome Project. Okay, now what is this Human Genome Project? Students, we all know, we were studying about nucleotides, we were talking about genes, we were talking about chromosomes, we even understood how our DNA, which is around 2.2 meters long DNA, is coiling around our histones, right? All that we saw. Now students, we need a data, right? We need a data to understand what went wrong, or if any mutation has happened, what went wrong there. To understand all of this, to understand the genome, to study the genome of a human, right? We needed a project. And this project was not anything smaller. This project was called the Mega Project. Okay, what was it called? As the Mega Project. Now, why was it called a Mega Project? Are you all with me? Are you all with me? Yes or no? Yes. Good job. Okay, students, this is called a mega project because many countries like Japan, France, Germany, all of these people came together at the same time. Many universities, many top institutes in the world which are renowned or which is known for, giving out the best content, right? When it comes to, scientific reasons, or when it comes to finding out something, when it comes to research, these people are the top people. All these people came together to make this a successful project, Human Genome Project. So first reason why I call it a mega project is because of the participation of many countries, right? Many countries took place or participated in this. So I can write many countries, right, participated. Yes or no? Yes, agreed all of you? Yes, China, Japan, we had a lot of people participating in this. The second reason why I want to call this a mega project is because they invested. Can anyone tell me how much did they put for this project? How much money was, taken into making this project a successful one? Can anyone tell me quickly? Yes, very correct. Shivakumar, to easily identify future diseases, that was one of the goals of them. We will study the goals of them also. Okay, we will understand the goals, but can someone tell me how much was put in for making this a mega project? Like how much, some amount was put in. Okay, I'll also write the years and everything. Okay, let's write who came up with the whole thing. Who came up with the whole thing? 3 billion, 9 billion US dollars. Very good, Rohini. Okay, very good. So students, it was James Watson who thought about this or wanted to bring this about, and they took a period of 990 to around 2003. Okay, it took around 13 years to come into picture, right? Look here, somewhere before I was born, from before you all were born, right, till 2003. It took 13 years to make this project a live one, so it could benefit us, so it could be of any benefit to us. Clear? Okay, it took around 9 billion US dollars. Okay, it took around 9 billion US dollars to make this a success. This is a lot of money. Can we even calculate the zeros? Too difficult, right? It is such a big money. So students, many countries participated, 9 billion US dollars. Not only that, Why it is a mega project? Because we are trying to understand the entire human genome project. That means we have to calculate for 3 into 10 to the power 9 base pairs, right? The entire human genome we have to find out. Everything we have to find out. We have to find the genes, we have to find how many repeateds are there, how many, how many tandem repeats are there, how many SNPs are there. We have to find everything about your human genome. So these reasons make it a mega project. Clear to all of you? Clear to all of you? Yes, very good. Okay, so this is the reason why it was called a mega project. Any doubts so far? And not only that, students, we should also add that if such a big thing, like such a big thing is happening, then there is a lot of people involved in that, right? We will need technicians, right? We will need technicians. We will also need a storage for all of this data to be stored. And they also found out that if I am trying to store this in a book pattern, right? Imagine you carrying an encyclopedia and walking every time and trying to find out, oh, maybe this nucleotide chain, that is why this happened. Oh, maybe this here mutation happened. Can you carry a book which is around 3,300 pages? Can you carry a book? Okay. Can you carry 3,300 books for each human? Right? 3 into 10 to the power 9 base pairs. So imagine each book has, okay, 1,000 letters. And same way it has 1,000 pages. So imagine you have to carry 3,300 books to understand what happened. Is there mutation happening? Why did this person get sickle cell anemia? Why is there difference in my hair texture? Why is my eyes like this? It's not possible. It's not practical. That is when bioinformatics came into place. That means with the knowledge of biology, human genome project, everything to do with molecular basis of inheritance. Same way using computer. Right? Yes, very good. Computer based project. That means we bring in computer and we also bring in the knowledge of biology and do khichdi. Okay? So that is how this whole thing. So this is just the introduction. Okay, what years, how many years it took, why it is called a mega project, what and all did we try to plan or find from this. Okay, now let's understand the goal. What is the goal of HGP? When they started this, now see, now 2024 started, we all have a goal that I'm going to be fit. That is one of everyone's goal. That is why all the gyms around this world will be full of people today, tomorrow, for next 1 week, because everyone will be like, yes, I am going to be fit, right? But after 1 week, no one will be fit. No one wants to continue the resolution. Students, why there is no likes? Are you people planning to bunk my class? Is this what you people have learned from New Year? I told you people you're not supposed to bunk class in such a young age. Okay, I'm going to check the attendance. Before that, quickly hit the like button. Come on. Show some love on New Year. I came on New Year, no, for you people. Good job, good job. Many of you have done the job. But more likes can come. Okay, more likes can come. Okay, so students, like that, how in 2024 when it was 12 o'clock or Jan 1st, you would have taken a diary, you would have written 100 things you want to do this year, you would have made a bucket list, right? Bucket list. But how many resolutions in that is going to cross 1 week of Jan? Only God knows. I don't think even God can figure it out, because even you cannot figure it out, right? So that is how this like when they start, they keep a lot of goals. Now we have to see how many goals did they actually accomplish, right? How many goals did they actually accomplish? Okay, let's see their first goal. NCERT lines. Okay, NCERT lines, same thing. Identify all approximately 20 to 20,000 genes. That was their main plan. They kept their resolutions very small. Not like I will lose 50 kgs this year, or I will lose 5. I will cut short my calories every day up to 500 calories I will reduce. They kept very minimal, good kind of resolutions, where they goals that was like identify all approximately 20 to 25,000 genes in a human DNA. Determine the sequence of 3 billion chemical base pairs. Correct? Your 3 into 10 to the power 9 base pairs has to be figured out. This was the one reason why it was also called a mega project. Correct? Now, store this information in databases. Here is when we brought in our computers. right? After attending school's computer class, I don't think students who have taken PCMB would have heard Charles Babbage, right? We have been talking about him all our life when we were in school. So here we are going to study about computer based, right? Computer based storing. Here is where we understood about bioinformatics. Bioinformatics. Students, actually this topic also lets you know that science, like people have a notion that science is a very, very constrained, there's no much options. If I studied science, or if I've taken PCMB, I do not have much options. Students, bioinformatics, it is amazing if you have interest to both computers as well as biology. Okay, bioinformatics is an option. Biostatistics is an option. Same way forensics. Today we are going to become CIDs and CBIs today in the class by trying to figure out who did what kind of crime. Right? So that time forensic comes into place. So all these things also are options. Right? So do not think like, oh my god, this year I couldn't crack NEET, so I have to take a big step of my life, or spoil my life. No, students, if you have prepared for NEET, if you have also written your boards, you also have a lot of knowledge. Go for, there are 100 other options. Agriculture is there, forensics are there, your bioinformatics are there, biostatistics are there. So nothing is the end. Okay, this is what I want to tell you on New Year. Do not think that this is the end of my life. Never. There are a lot of people who tried to find out something else and ended up finding other things, right? We have a lot of scientists whom we keep studying daily basis. He was trying to do about pus cells and he found out nuclein, right? Same way our Altman found out the acid content. Now we all study about him, right? So things happen for the best. Trust the process. Okay, fine. Now computer based bioinformatics. Improve tools for data analysis, because here when we talk about your HGP project, it is lot about data. Okay, it is a lot about data, because you are actually cutting short on 3,300 books by making this a computer based project. Otherwise each person should have had this book now, right? By making it a computer based project. Students, I don't know how many of you have noticed, if you sometimes see in some places, okay, in some places you will see those people are very, very, very prone to have diabetics, or those people are very prone to have PCOD. So people will be like very, suspicious, like why only our state viral disease, or why do now if you notice Kerala, lot of now the COVID variant one, or if you see Nipah virus, all these things have very prone to come to Kerala first or get affected in Kerala. Mainly it could be because it is a coastal region. Other than that, there could also be as an entire as an entire state, it could be that their genome or their SNPs are modified in such a way that they are prone to this kind of a disease, okay, or they are prone to this kind of a mutation. It is lot in depth, let's not go there, but just understand, okay, that also can happen. Fine. Transfer related technologies to other sectors. Okay, so students, understand, whenever we are doing something like this, like I just told you about Kerala situation, or I'm telling you about places, some places are there, imagine some kind of disease have outbursted in that area. We are trying to figure out 100 reasons why it would have happened, but we are not able to find out. Understood? We are not able to find out. Thank you, Mr. Cool. Very cool of you to type that. Okay, so students, what is happening? So when such a thing come, will you be able to go and refer in this book, oh, this person's here, it would have changed because of this mutation would have happened? No one has the time for that, right? No one has the time. We need a solution. If I am trying to figure out that, see, AUG will code for methionine, I need this data. I cannot go search in this 3,300 books where this is written. I cannot find, right? So that time what happens, transfer related data to whichever particular sectors are there. I want to transfer it to a sector where the doctors are trying to find out a vaccine, or I want to transfer it to some industries which are producing vaccine. Anything for that matter, I should be able to transfer the data. Clear? And the last one, very, very important point, okay? Many times questions has been come from this one, full form. I have also mentioned it prior also. Students, look here. Yeah, Doctor Strange, many days over. Now I'm seeing you. Yeah, I saw a lot of subscribers on my personal channel also. I'm like, students, why? But it's nice of you people to think like that. Okay, students, look here. Address the ethical, legal, social issues. Okay, what do they mean by that? Okay, this full form has been asked many times. LC. It's a girl's name, so you people shouldn't forget. LC. Okay, LC is the person whom you shouldn't forget, because it's a woman you people shouldn't forget at all. Okay, so ethical, legal, social issues. I also is important. Issue also is important, right? So this LC, that means what, students? Now I know, imagine. Okay, now I know the genome of everyone. Okay, I know the entire genome of every single human. Easy for me to make a clone. Yes or no? Yes or no? Easy for me to make a clone. I can make a clone of some actress. I can make a clone of President of India. I can do anything, right? So that also should not affect. Just because we are working on an entire genome, it is easy for us to make a clone. So that will come into ethical and legal issues. There should be legal issues tied up with it. Otherwise, every person will get up tomorrow and they'll be like, okay, I'm going to make a clone. Right? That should not work. That should not happen. So, and knowing this genomes, this genome should not also affect the society as a whole. No one should be able to, be easily to alter or create a mutation by themselves, right? Like how we have bio wars. Social issues also should not be there. Clear? Social issues. People who are missing issue will have to have a tissue in your hand after NEET exam, because you will lose 4 marks. Okay? Those who forget the word issue. Right? Yes or no? Haha. Yeah, you people will make clone only, no? Yes, ready? Understood? Issue word you shouldn't forget. Most of the students will be like, who will remember issue one word? But then LC's last word I stands for issue. Okay? Yes. Okay, now let's understand what are the ways. That means, students, now you understood that this is how I have to do it, or this is the issues behind it, right? Issues behind it. Now I have to understand the methodologies. Okay, methodologies. Now what do I mean by methodologies? I am talking about how do I do this. Now goals are set, right? Your goal was to, okay, all of you tell me what was your resolution this year. All of you tell me in the chat box what resolution. Only one resolution. One resolution, you put it up in the chat box. Let me see what's your resolution. And we will take one. I will pick out one example and I will tell. Tell me. What is happening? So much fun they are having among themselves. Okay, crack NEET. Okay. Other than cracking NEET, is there anything else that you people have kept as a resolution that, okay, this year I am going to do this no matter what? No matter what. I think chats are running slow here. Let me check in my phone. To crack NEET, to eat to eat every day. There are people who do photosynthesis, they have decided to eat every day instead. Wow. To get replies. And you're getting my reply. My presence on a live is the reply. Correct? Why you want more reply? Why are you feeling sad about it? Okay. Read 5 books. Okay. My resolution I have already told in that NEET video, you should go watch. Okay, fine. Most of you is crack NEET. Let's take a person who is decided to eat every day. That is Janice. Okay, to eat every day. Very simple resolution. All this while they were doing photosynthesis, so now they have decided, okay, I have to start eating. Right? Students, you have to eat for a healthy life, right? So what is happening, students? What is happening? Here, methodologies when I talk about, they set the goals. Same like, okay, I have to eat every day. Now for me to eat every day, I need to wake up in the breakfast time. I need to wake up in the lunch time. I need to be awake in the dinner time, right? If you're postponing and you're waking up at 1 o'clock, then you will eat brunch. You will eat lunch and breakfast together, right? After that, you feel like, okay, I will sleep for some more time, and dinner time also you skip. Then you will not be able to eat, right? You will not be able to eat, okay? So that is the reason also, students, understand that when you're setting something or when you're setting a goal, you also need to know how you are going to, make that goal. All of you have told crack NEET, definitely that is your goal, okay? Crack NEET is your goal. Now if you want to crack NEET, what are the methodologies that you follow? That is what this HGP also done. Very similar, your HGP and your lives are very, very similar, okay? So let's look here. First methodology, students, very important. Two methodologies they did. First one is called as sequence annotation. Sequence annotation. And second one is called as expressed sequence tag. Yeah, express sequence tags, ESTs. Okay, all of you have your NCERT? Students, most of my groups where conversations can happen is completely stopped. Okay, I will not be texting, nor you can text, okay, because of some disturbances, right? Some unnecessary comments, that's the reason. So you will have to wait for some more time. After that, all of you will be able to text and let me know if you have any doubts. Okay, Zen? Yes. Students, okay, listen here. First one is called sequence annotation. Next one is called express sequence tag. I want all of you to keep your NCERT open. Open the paragraph which speaks about methodologies. Okay, which speaks about methodologies. Clear? Yes, very good. EST. Rohini is very active. Others are all chatting with themselves. Okay, students, so in sequence annotation, what happened? We did something called as Sanger sequencing. Students, nothing, very simple. There was a person called Sanger. He did something called as DNA sequencing. That means the entire DNA sequence, AGGC, okay, all that AGCT, all that sequence, right? He was able to, get it properly or to sequence it properly. Okay, so that is why. So when you did sequence annotation, we did first one is called as Sanger sequencing, where we did not care if it was going to be coding or non-coding. We just took the entire sequence. All of you know, in our DNA, in our gene, there is one portion which is called the introns and other portions which is called the exons, which we understood in our translation, transcription when we did, right? So here in Sanger sequencing, we did not bother if it was coding or non-coding. We just took the entire sequence. Okay, that was the first methodology they took, because here we are trying to find out, right? Here we are trying to find out what? Here we are trying to find out the entire genome, what is useful, what should be known to us, how to understand where mutation could happen. Okay, so students, in Sanger sequencing, we got both. What and all did we get? We got coding sequence and non-coding sequence. Clear? Coding and non-coding. Important, because in some places when we are doing methodologies, we only prefer coding. Okay, that is what you are going to see next. Yes, are you all with me on this? And they also had, other than DNA sequence, other than coding and non-coding, they also found out amino acid sequence or protein sequence. That is a very, very plus point for this. So when they studied the DNA, so students, simple, you can say it like this: sequence annotation, they took DNA, they studied the DNA with the help of Sanger sequencer. They did Sanger sequencing. It is called as DNA sequencer. Okay, the entire setup is called as DNA sequencer. With that, because the person who found it was Sanger, we call it Sanger sequencing. Sanger sequencing, we understood, we took a DNA, we studied it, we understood coding and non-coding is that we sequence that. Other than that, we also sequence amino acids. Amino acid sequence. Clear to all of you? Are you all with me? Yes? Okay, great. Good job, good job. All of you? Fine, great. Students, 1 minute, I'll just get some water. Yes. So, important concept, do not be distracted, try to focus. This was the first methodology, which is called as sequence annotation. Now let's understand what is express sequence tags, like what is the difference. Okay, students, in express sequence tag, they focused on taking only the coding strand. What did they focus on? Taking the coding strand. Now focus here, like try to understand this. When you want a coding strand, we have all understood, we have to extract it from our mRNA, right? So what we did, we have to extract from our mature mRNA. Now what do I mean by mature mRNA? The mRNA that we got after we did splicing, after we added a 5' and a 3' end, after we did capping and tailing. Is this clear? Yes? Clear or not? I'm doing great. Thank you so much, David. Students, so we extracted the mRNA. I want all of you to understand this. This mRNA is what? It has only the coding sequence, right? Because we removed the non-coding part. We removed the introns with the help of splicing. Now, with the help of this mRNA, I'm going to do reverse transcription. What am I going to do? RT, I'm writing reverse transcription. Now, when I do reverse transcription, am I not going to get my DNA? I'm definitely going to get complementary DNA. I'm going to get complementary DNA, correct? Now this complementary DNA and this mRNA will come together, right? They will try to form a bond. Okay, now they will come together. They will try to bind with each other. Now, I do not want mRNA. I only want DNA, because what am I going to do? I am doing methodologies. I need to find the genome. My job is not to bother about the mRNA. I use the mRNA so I can get a coding strand. I need a DNA which has only the coding. I do not want non-coding. Clear? Okay. So, now what am I going to do? With the help of, students, all of you tell me, if I want to digest this RNA, if I want to digest this RNA, what can I use? What can I use? We have seen this in Avery, McLeod and McCarthy experiment. Can anyone tell me? Thank you, Janice. Can anyone tell me, if I want to digest this mRNA, I can use an enzyme. I can use an enzyme. I just want that. Quick. Answer should come very fast. No, not nuclease. The name is there here only, students. Just look at it. If I want to digest this RNA, right? I have to digest this RNA. What do I need? Very good. Who told the first? Prashant, Rohini, Pranav, Dinesh, YS. Very good. Okay, students, I need to use an RNase, correct? I need to use an RNase. Let's go to the, 1 second. Yes, okay. Students, so I'll write it here. Okay, so for your ESTs, okay, EST. Yes, for your ESTs, we took one mRNA, okay, one mRNA. That mRNA Oh, yes, it's visible, right? Okay, that mRNA, with the help of reverse transcription, we made a cDNA. Now this cDNA and this mRNA is going to come together, which we don't want. Okay, but still this will happen. After that, with the help of, how all of you told, with the help of RNAs. Okay, with the help of RNAs, I will be digesting the mRNA. Okay, I'll be digesting the mRNA, which will leave me only with the cDNA. Okay, only with the cDNA. Now what do I need? I need one more strand, correct? I need one more strand of DNA, right? For my bonding to happen, we need double strands, right? So with the help of polymerase activity, that means using my DNA polymerase, activity, I am going to get 2 strands of DNA. 2 strands of DNA. Got it, all of you? Now all of you tell me, this DNA, right? This DNA, will it have coding sequence or will it have non-coding sequence? As a whole, will this have coding or non-coding sequence? Now we took the mRNA, we formed DNA. Now all of you tell me. Who are the beautiful spammers? Yes. Is it coding or non-coding? This DNA which I am going to get, will it have coding or non-coding? It will definitely have what? Very good. It will have coding, right? It will only have the coding sequence. So you understood how did they find? Very good. Okay. So this is how their second step worked. What was it? Express sequence tags, where they used only coding sequence. And how did they form the coding sequence? They took a mature mRNA, they did reverse transcription, formed a complementary DNA. From that they used RNAs to remove the mRNA and used polymerase activity to form a 2-strand DNA, which will be having only the coding sequence. Very good. Okay, clear, easy. This is how they did expressed sequence tags, and sequence annotation was a complete uniformity. They took everything, coding, non-coding and amino acid sequence. Okay, so look here what your NCERT says. Okay, we will also write 2 things, very important. I'll write it here only, students. All this has to take place with the help of a host and a vector. So the host used was bacteria. This is only for your notes purpose. Okay, bacteria and yeast. And the vector, we understood vector, host, all this when we were studying chikungunya, dengue, right? So vector is back and yak. That means bacterial artificial chromosome and yeast artificial chromosome. Okay, clear? Yes, great. Okay, so now look here, this is your NCERT lines. Nothing, everything is similar. The methodologies they used express sequence tags, okay, and they used sequence annotation. For sequencing, the total DNA from a cell is isolated and converted into random fragments of relatively smaller size, and here brackets are, and cloned into a suitable host. Okay, they used a host for cloning. And what was the host that they used? It was neither bacteria or yeast. And how did they clone it? With the help of a vector, which is a yak and back. Okay, using a specialized vector, the cloning resulted in amplification of each pieces of DNA fragments so that it subsequently could be sequenced with ease. Okay, amplification or increasing or making into fragments or isolation, all these techniques are used just to make it comfortable for us. Okay, the commonly used host is your bacteria and yeast, and the vectors are yak and back. Clear? NCERT lines also clear? Okay. Here you can see the other took a blind approach. So here first they are talking about your express sequence tag. Next, they're talking about sequence annotation, where it is called a blind approach because they did not care about the coding or non-coding. They took the entire sequence, including the amino acid sequence, and they just made that kind of a methodology. But when it comes to express sequence tag, they were very specific. That is the reason why sequence annotation is called a blind approach. Clear? Okay? Yes. Now, since we understood how the methodology, there is no in-depth about the methodology, but understand that they took the DNA, they sequenced it, they took the coding, they took the non-coding, they separated everything, and they were trying to understand the genome. Now, what are the salient features of the HGP project? The human genome contains 3164.7 million. Students, this you have to memorize. There is no other way. Understanding will not help because these are all numbers. Okay? Clear? These are all numbers. Students, I would request you people to not spam, okay, because you people are disturbing unnecessarily. Yes, it is also disturbing for others, so I would request you people not to do it. Yes, fine. Okay, students, look here. This numbers, no explanation, just you have to understand it by heart. Okay, 3164. The average genome consists of 3000 bases, but the size varies greatly. That means an average genome, the average gene, okay, it has around 3000 bases, but gene size can vary, smaller, bigger, right, larger one. So, but the size vary greatly. And the largest known genome is your dystrophin, which is around having 2.4 million base pair. That means what are they trying to say? Average genome, okay, average gene will have around 3k base pair, 3000 base pair. But the largest one, that is your dystrophin, will have around 2.4 million. Okay, this dystrophin is a protein based gene. This actually helps in your muscle, like, you know, for flexibility of your muscle, for that rigidity of your body, and it also helps in survival, like for your longevity of life. Okay, how long a person will live, all this can help. Okay, dystrophin can help, and lack of dystrophin will cause, again, your syndromes also are there. Okay, that also will be caused, which will be studying again in your biotechnology. Okay, Duchenne syndrome. Okay, students, look here. The total number of genes is estimated to be 30,000, much lower than the previously they thought the genes were around 80,000 to 1,40,000, but then actual HGP project gave a clarity that it is only 30,000 or much lesser than that, not more than that. Clear? Again, you have to memorize. The functions are unknown for over 50% of discovered genes. Can you believe? Around 50% genes only we know. What it is doing, where it is doing, what is happening, but the rest of the 50 we don't even know. We don't even know. Okay, so the rest 50 is not it. Students, this point is very, very important. Look here, less than 2% of the genome codes for protein. Now, can anyone decode the statement and tell me? Can anyone decode the statement and tell me? Okay. Where you have, see, this is million base pairs. If you have a confusion, just go for this. Write 3 into 10 to the power 9 base pairs. Okay, just write this. Do not write million, billion if you have a confusion. Okay, clear, Rohini? Do not write million, billion if you have a confusion. Okay, if you see in this whole thing, everywhere it is million, 2.4 million. Okay, yes, fine. Students, look here. Less than 2% codes for protein. That means 98% is junk DNA. That means DNA which does not, it's non-coding. And only 2%, right? Only 2% or less than 2% is your coding sequence. What does this mean? We have more junk than what is useful, right? This 98% is actually your junk, which is not doing any coding for protein. Okay, very important statement. Okay, very important statements. 98% junk, not doing any coding, not helping you in sequencing, not helping you to understand, oh, this is how my texture looks, protein, this protein will code for this one. Nothing it's doing, junk. 2% or less than 2% is actually coding. Clear to all of you? Clear to all of you? Yes. Very good, very good. Someone decoded it. Who was that? Muller. Muller decoded it. Non-code. So, only the coding. 2% is coding for protein. This is the only 2% that is actually helping for the formation or sequencing of your protein. The rest is full non-coding. Now you will be like, ma'am, then why should I study about this? Why is this 98% even there in my body, right? Students, this 98% will help with identification, will help with lot of sequencing with other jobs, right? It's not like everything is useless, 100% 98 is useless and only 2% is useful. No, 2% is doing your translation. 2% is being able to have the ability to code for a protein. Others are not having the ability, okay? Now, repeated sequence make up a very large portion of the human genome. Now, what do I mean by repeated sequence? That means in a person, there's something called as tandem repeats, which I will teach you now. Okay, there's something called as tandem repeats. Now, let's say 2-3 names: Pranav, Shena, and Rohini. Okay, 3 people's name I've taken: Pranav, Shehnaz, and Rohini. Okay, these 3 people are there. They are the heroes and heroines for today. Okay, students, so Pranav's tandem, students, Pranav's tandem repeats is say 10. Okay, some sequence, let's say ACTG. Okay, ACTG is there in all of them, but in Pranav 10 times this is repeated. Okay, in Shehnaz 25 times this is repeated. In Rohini, maybe say 2 times it is repeated. Understood? Okay. So, what does this mean? What does this mean? That means this tandem repeats for Pranav is not going to change. Not going to change. You take his hair, you take his blood, you take his saliva and do test. Still it's going to be 10. 10 tandem repeats. That means that repetitive sequence, this number will not vary. It will be 10 itself. Same way for Shehnaz, it's going to be 25 itself. Okay, Shehnaz also it's going to be 25 itself. No matter you take her hair, blood or saliva, it's going to be the same. Going to be the same. Okay. Okay, Praveen. I've been telling Pranav, sorry. Okay. And for Rohini, it is going to be 2. That means this ACDG sequence is going to be repeated 2 times. Tandem repeats, how many repetitive sequence she has? 2 times. Clear to all of you what tandem repeats are? Tandem repeats are clear? Yes? Okay, great. Now, look here, these are again numbers that you need to know. Chromosome 1 has the most genes, which is around 2968. Okay, 2968. And Y has the fewest, that is around 231. This also has the smallest gene. Okay, this also has the smallest gene. It has around 14 base pairs. Okay, and this is also called as TDF, which is your testis determining factor. Okay, this is also called as your testis determining factor. Okay, clear to all of you? So your Y has the fewest genes, and it's not only the number is fewest, it also has smallest genes. It might have around 14 base pairs, and it is also called as TDF, which is your testis determining factor. Okay. Now, scientists have verified this is your SNPs, which I'll be talking about. Okay, let's talk about SNPs as separate topic because that has so much to do with our life. Okay, students, SNPs or known as single nucleotide polymorphism. Okay, now I want 3 students to volunteer and tell me, I will take your names. 3 students, only 3 students I want. 3 students. Come on, just tell me, ma'am, me. How you tell no in class? Ma'am, me. We tell in school, no? When the teacher calls. And that who will take care of the class? Okay, Dinesh. So much time to tell your own names, huh? YS. Zen. Okay, I have taken 3 names. Other names we will take later. Okay, next class. Okay. I think. Okay. Students, we have Dinesh, Zen and Devika. Okay, Dinesh, Zen and Devika. Okay. So, these 3 people. Students, if I am going to tease you people, you shouldn't be offended or you shouldn't be like, oh, Gopika ma'am teased me and all that. Don't cry. Okay. I'm just making other students understand with your names as examples. Yes. Students, in your Super 6 series itself, where else it will be completed? In Super 6 series only. Super 6 series only. Okay, okay, fine. Let's write Zen's, okay, sorry, let's write Dinesh sequence. Okay, this is just to make you understand. G, C, T, A, G, C, G, C, G, C. Okay, let's write Zen's, okay, G, C, T, C, G, C, G, C, G, C. Okay, and then we have Devika, again G, C, T, A, T. Okay, G, C, G, C, G, C. Okay, now we have 3 people in our class. Okay, 3 people in our class. We have Dinesh, Zen and Devika. This 3 looks very, very different, right? This 3 looks very, very different. Their behavior, character, personality, everything is different. But just look here. But look here, what makes them so different? Look here, all the sequence almost are the same. But if you look here, you can see this. Can you see? Can you see? In Dinesh, A, okay, adenine has come. At the same place for Zen, cytosine has come. In the same place for Devika, thymine has come. Clear to all of you? Clear to all of you? Yes or no? Yes. So, students, at the same place, the rest of the entire sequence is the same. Only this sequence ACT has changed, which makes them very, very different. It will be like that one nucleotide could be the reason why I have a straight hair. That could be that same position, maybe another nucleotide would lead to another person having curly hair. Right? Got it, all of you? So that one change in a nucleotide or one change in that base pair causes a lot of difference. So this is called as SNP. Single nucleotide polymorphism. That means because of a change of a single nucleotide or change of a single base pair will lead to a complete change of an individual. Okay? Yes, good job. Now this SNPs, how many SNPs do we have? We have around 1.4 million SNPs in our body. Okay? 1.4 million. Are you all with me? Yes, good. Okay. Understood? Yes. Now students, this 1.4 SNPs doesn't mean it should be only in the coding sequence. Not necessary. It can be in non-coding or coding sequence. It can be in non-coding or coding sequence. It can be anywhere. There's no necessity that it should be only in the coding sequence. It can be in the non-coding sequence. It can also be in the coding sequence. Because I told you, even though that DNA is junk, it helps in identification. It helps in variation. It helps in many other sequencing. Okay. So this much is clear to all of you. Now students, can I always say that, SNPs are always beneficial? Not necessary. Sometimes, like how we have, you have HBS, that means your sickle cell anemia. Okay, sickle cell anemia. Okay. Students, in sickle cell anemia, if you have noticed, it is caused because of SNP. It is caused because of SNP. That means one nucleotide changed in a person, due to which that person got affected with sickle cell anemia. Got it? Sickle cell anemia. So it's, and also there is also instances where your sickle cell where your SNPs are also of benefit. Okay, there is CCR5. Okay, CCR5, it is actually one base pair change because of which that person became, or that entire genome became resistant to HIV. So it's not like always SNPs will be causing you any kind of mutation or diseases. There are cases where SNPs can also be beneficial, like how I told you, it became resistant to HIV because of the change in one base pair. Okay, so like that instances are there, but also understand it's because of SNP that sickle cell anemia is caused. That means that one nucleotide is enough to cause a big trouble as well as could be a benefit. Okay, yes, great. All of you understood. All of you understood. Students, did you understand so far? All clear so far? Yes, no? Okay, now we will understand what we spoke about repetitive. Repetitive DNA, what we spoke about. Students, repetitive DNA, I can have it two ways. Now what could be the repetition? Now say I take whose name did I not take? Shana's name I took. Everyone's name I took, no? Okay, so students, look here. Two sets of repetitive DNA can come. We understood that repetitive DNA are around 98%. This repetitive DNA are there, and they are only present in the non-coding sequence. They are present in the non-coding sequence because the coding sequence will code for protein, and that is around 2%. Okay, now imagine I write one kind of a sequence: ACTG, 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 AA, ACTG. Okay, AA, no, TTA. Okay, I want all of you to focus what I have written. Okay, look here, all of you. I have written two kind of sequence. Okay, two random sequence. Okay, now imagine one is Kim sequence, one is Muller's sequence. Now this is just an example. Okay, both the sequence can occur in any kind of a person. Okay, there is nothing like only this will occur, only that will occur. Nothing like that. It is there in our body. Both kind of sequences are there. Now students, look at the sequence ACTG, ACTG, ACTG. This is a kind of continuously repeating sequence, right? This is a sequence where there is a continuous repetition. Look here, ACTG, TTAA, ACTG, TTAA. That means they have gaps, right? They have gaps. Between ACTG, TTAA has come. Same way between TTAA, ACTG has come. Gap sequence, right? This is having some kind of gap. Clear to all of you? So repetitive sequence can be continuous, or it can be with the gaps. Clear? Yeah, they are regular spammers. They have dedicated their time to come and every class and message each other. We should respect such kind of people, okay, who have so much dedication. Yes or no? We should respect regular spammers. Are you all with me? Just copy down the sequence. Copied? So this much you have to understand. This portion will be continued in your next DNA fingerprinting. So just understand this topic, that this could be continuous or this could be gapped. Okay, now let's understand DNA fingerprinting. Okay students, in DNA fingerprinting, when we talk about generally about DNA fingerprinting, it's mainly, okay, let's write here, DNA fingerprinting. Now imagine a crime scene happened. Okay, a crime scene happened, and that criminal who wanted to murder a person happened to feel thirsty. Okay, most of the criminals will not feel. This is a very made up story by me, that the criminal felt thirsty. Okay, so the criminal felt thirsty. He touched this glass. Okay, he touched this glass. Crime scene. He forgot to take this glass while leaving out of the crime scene. Okay, now what is going to happen? Forensic team will come. They will start investigating everything. They will notice that there is fingerprint on this glass. Okay, now usually the fingerprint could be because of the person, the criminal itself, or the person who was murdered. It could be any of them who drank the water before being killed or before planning to kill a person, right? But they will take this glass and go without making their fingerprint on this. They will be all wearing blouses. They will take this glass and go. Okay. Right, they'll take this glass and go. They will check the fingerprint. Now, since it is fingerprint, what happens, students? Normally fingerprints at some point of time actually fades. Okay, it is because of the excessive, we use this hands a lot of times. At the same way, a person who is sweating, right, a person who always sweats, there are chances that the lines become, fade. The line starts to fade away. So we cannot extremely depend about fingerprinting, but we can also, because now we know that using our fingerprints, all of our biometrics like your Aadhaar card, PAN card, everything is connected. So if there is a chance that the fingerprint is there in this crime scene and it is Gopika's name, definitely they will come and investigate on me, and they will understand my alibi, where I was on that day, what I was doing, what do I have connection with this person who died, and all that. Correct, true. But there are situations where people also modify this kind of fingerprints, and there are criminals. Now if you have seen documentaries, the criminals are very, very smart. They wear gloves and come. They wear bikers, riders, gloves and come. They do all of that, right? They maximum make sure that they do not allow any fingerprints in the crime scene. Now in that situation, what do a person do? What do the forensic people do? Do they let the crime happen? Do they be like, oh no, I cannot find out anything, so I will just let the person kill a person and go? No, that is not possible, right? So in those situations, students, what happens is, so DNA fingerprinting, let's understand this quickly. Okay, yeah, okay, so in these situations, what happens? Imagine we got some kind of DNA fragments. Now how did I get a DNA fragment? I would have found a hair in the crime scene, or a saliva, or a blood, right? Most of the time when a person is trying to kill if they have not made them unconscious, they would have scratched this person. Because of that, in the nail they would have found some blood samples. Lot of cases have been solved because of that. So anything can happen. We somehow got some amount of DNA fragments, right? Now students, we understood from, because you are a biology student and I am a biology teacher, we have understood that 99% of our DNAs are the same, and this DNA is called the bulk DNA. That means for every single human, actually 99.9%, okay, 99.9% DNA is the same, bulk DNA. Now what makes you and me different is this 0.1%, which you call it as a satellite DNA, okay, or people also call it as light DNA, main DNA, many words you can see for this, okay, main DNA. Okay, all these terms you can see in different places. If you check, you will see different words. Now students, what are they trying to say? Me, Rohini, YS, Zen, everyone has 99.9% same DNA, but we are very different from each other. Even though we have not seen each other, we are very far from each other, we are still having 99.9% same. What is making us different? This 0.1%. This 0.1%. Clear to all of you? Now, when I am trying to investigate a crime scene, should I focus on this or should I focus on this? Should I focus on the bulk DNA or should I focus on the main DNA? All of you tell me. All of you tell me. Should I focus? Very good. I should focus on the satellite DNA or the 0.1% DNA. This makes me easier to solve a crime. Yes? This makes me easier to solve a crime. Okay? Now, this was actually first figured out by a person called as Alex Jeffrey. Okay, he was the person. He's called as father of DNA fingerprinting. Okay, Alex Jeffrey was the person who first figured this out, and he's also called the father of DNA fingerprinting. Okay. Now crime scene, things are happening. I got a DNA fragment. Now what is my job? I have to make sure that I'm not checking on this 99.9%, I'm checking on this 1%. So I'm going to use, restriction endonuclease or your biological scissors to cut off whatever is not required for me. Correct? Whatever is not required for me. If satellite DNA is absent, then we all would look, no variation at all. We all would look like clones. We all would look like how bacteria reproduces, how asexual reproduction would have happened. We would have looked exactly like that. Okay, so students, what did I say? Now imagine we took this DNA fragment. Okay, I cut with the help of restriction endonuclease. I'm writing RE, restriction endonuclease. Okay, I'm cutting the fragments. Cut into fragments with the help of RE. Okay, restriction endonuclease. After cutting into fragments, I will run it with the help of an agarose gel. Now, what are we talking about? We have taken the fingerprint, we got a suspect or we got 2-3 suspect. These people have manipulated the fingerprint. It could be this glass would be bought from some other place with the fingerprint by a criminal just to make sure that the police, deviate from the case. All this have happened. Many times all this have happened. So, say we got a person, we got a person who had the same fingerprint. But that does not prove the crime scene in 100%. We have to find something. We have to find something that has a DNA fragment, or we need to have other evidence like a CCTV footage from a person entering a crime scene, all that. But then all this is not bothering us because we are not computer students, we are biology students. So we are only focused on DNA fragment, right? So that also done. I will run it through an agarose gel. Okay, I will do AGE or polyacrylic, that is page. I'll do age or page. Okay, I'll do age or page, by which I will understand that this 0.1% which this fellow has got from his maternal and paternal will not coincide with any other person. Correct? Whatever 0.1% is there in me will correctly be shown in this agarose gel. Because what is happening in agarose gel? Density gradient, centrifugation, like how you have centrifugation, density gradient, same way bands will be shown. Okay, which I'll show you in this image. Okay, so by then I will understand that okay, this fingerprint was misleading. Actually, the criminal is a different person because I got hair sample or blood sample from it. Okay, yes, very good. Agarose gel. Okay, and students, now if in case, imagine before going to agarose gel, I want to do a centrifugation. Okay, with the help of a density gradient. Now, can any of you tell me what did we study in Messerson and Stahl experiment? Density gradient with the help of a centrifuge. Okay, with the help of a centrifuge. What is going to happen? I am going to get 2 strands, correct? Now we know this. We know this. Imagine we are going to initiate this process. I am going to get 2 strands. Right, what are my 2 strands going to be? One is going to be here and one more is going to be here. I am going to get one heavier, one lighter DNA. Heavier DNA will be what? This 99.9%. Lighter DNA is going to be what? This 0.1%. All of you agree? Yes, very good, Malar. Cesium chloride. right? When we did density gradient in Messelson and Stahl, we did cesium chloride we used as a density gradient. That means just to understand which is the heavier part, which is the lighter part. Now heavier part will be your bulk DNA, lighter part will be your 0.1% DNA. No doubt in this. This is a way, I'm telling you, this is a way to understand. Students, even in identical twins also, if you notice, there will be some kind of a change. There will definitely genetically be some change. It could be in their hair texture, it could be in their eye lens, it could be the slightest of slightest thing which an SNP could have changed, which is not visible for us, which we will not understand just by looking at them. Right, there could be something that is different, okay, something that will be very, very different. Their fingerprints are different, right, their fingerprints are different, correct, Jyoti? When their fingerprints are different, otherwise both of them can have same Aadhaar card, no, fingerprints are definitely different, like that. Texture wise, you know, small variations will definitely happen because of your SNP. That SNP is enough for us to understand if this person did it or the other person did it. Clear? Yes, E. coli and all we use, no? So this could be one way of understanding how I have a bulk DNA and how I have a light DNA. Now, if I'm doing DNA fingerprint, let's understand what are the steps I would take. Okay, now you are a forensic person. We have entered a crime scene. We have 3 suspects. Okay, we have A, B, C. Do anyone wants to become suspect? You can volunteer to become suspect, but later don't complain. Okay. So A, B, C, 3 criminals we have. Now imagine we got DNA samples of any 2 people, or we got DNA samples of all 3 of them also. Okay. Now what will I do? I will isolate the DNA. First my step is isolation of the DNA. Now isolation means what? I am taking the saliva sample, I am taking the blood sample, I am taking the hair sample and keeping it untouched. Have you seen forensic people? They will use forceps, they will take the hair, they will put it in a plastic bag, they will seal it, right, and they will send it to the forensic department. They will send it directly to the labs. They will not be checking it, oh, hair sample SNPs there. No, they won't do that, right? They will send it to the laboratories where the people are actually doing. So a lot of students are there who do MBBS and take specialization in forensic and go, but then you need to have a very lot that, you know, mental capacity to see kind of crimes that is happening nowadays. It's very difficult to see, right? So that much of willpower you should have for that. Okay, so isolation is done. Now what am I going to do? Digestion of DNA by restriction endonuclease. That means, students, what? We are going to remove the bulk DNA and we are only going to focus on the 0.1% because we need to find who this suspect is. Is it A, B or C? Right? Now, after that, what am I going to do? I'm going to separate the DNA fragments by electrophoresis. Here is where I will use age or page. Okay, age or page. That means agarose gel electrophoresis or polyacramide agarose gel electrophoresis. Okay, yes or no? Clear to all of you? Then what will I do? I got age or page, like I got the electrophoresis. Now how does electrophoresis work? I told you. Now imagine, where do I draw it? Here I'll draw it. Okay, imagine this is your agarose gel. Okay, you can see it in the next diagram that I add also, but otherwise I'm telling you, students, DNA is negative, so it runs this agarose gel or this electrophoresis. We will learn in depth in biotechnology, but just understand this works on the principle of charge. Okay, charge and energy. Okay, that means charge and length, band width, width we tell. So that one. So students, what happened? DNA is negative. So if I keep something positive, right, DNA is going to run towards the positive. So if the bandwidth or if the fragments are lighter, they are going to run faster. faster. A person is lightweight, they will run faster. A person is a little healthier, the person might find a difficulty to run. Same way, here a lighter person, I'm not generalizing, there are a lot of people who run otherwise also. Now imagine, just telling, lighter DNA fragments will run faster and form a band there. Okay. Same way, other people also will start slowly forming bands. Okay, they'll form bands. Okay, many different types of bands will be formed. This depends on positive and negative charge, because DNA is negative and it will run towards positive, and if they're lighter, they will run faster and reach first, forming the band on the top. Then slowly the heavier particles will come and settle in the bottom. Okay, clear? So that is how your electrophoresis generally works. Lot you will study in biotechnology. Now transferring this, now I got this. Will I carry the gel electrophoresis and walk around? No. I will transfer it to your blotting. It's called as southern blotting. There is northern blotting, western blotting. Okay, which will be different. You don't have to know so much. But understand, this type of blotting is called as southern blotting. Okay, that means you're taking this gel electrophoresis and you're making an impression. You're making a copy, so, which you can walk around with, which you can carry around with. Okay. Transferring or blotting of separated DNA fragment to synthetic membranes such as nitrocellulose or nylon. Okay, these are the two membranes that we are using. It will be made up of nitrocellulose or nylon. Okay, nylon sheet. Okay, this process is called as southern blotting. Southern blotting. Southern blotting, where all of you are understanding? Is there any student who hasn't understood anything? Very simple, isolated, digested unnecessary things which are not required for us. We are focusing on 0.1%. Then you are separating the DNA fragments depending on a gel electrophoresis which runs on a positive, and the bandwidth which is lighter will run faster and make a band. The other ones will settle below. Okay, then what am I doing? I cannot carry this gel electrophoresis and walk around. It is a whole setup. Movement will cause difference in the band. So I will transfer this gel electrophoresis to a blotting paper which will be made up of nylon or nitrocellulose. Are you all clear? Can I see some fire emojis in the chat box? Very good, YS. Now, hybridization using labeled VNTR probe. This I will teach you now. Okay, hybridization using a VNTR probe. Now, this VNTR probes will be radioactively labeled. I had taught you last. Okay, I will tell you again. Now imagine, student, that 0.1%. Now imagine A is the suspect. Okay, A is the suspect, and I got a sequence which says ACGT. Okay, this is what the sequence I got. Okay, this is the sequence I got. Now my VNTR probe, right? My VNTR probe will be, what is A complementary T, C complementary G, G complementary C, T complementary A. This is my VNTR probe. That means which will go and bind with this one. Got it, all of you? Now ACTG, imagine, is only present in this A person. This A person. When I send a VNTR probe which is TCGA, CA, where will it go and bind? To this A person's gel electrophoresis. Right? It will correctly go and join on the fragment which has this. Agreed, all of you? Got it? Yes? That is your VNTR probe. That means a complementary fragment or a complementary sequence to your satellite DNA. Complementary sequence to your that 0.1%. Right? Okay. Now, detection of hybridized DNA fragments and by autoradiography. I told you this will be radioactively labeled, this VNTR probe. Because of that, I will be exactly able to see which is the one that is correctly gone and bind. Right? Which is that 0.1%. In which person? Is it in A, B or C? Who is the criminal? Who is the ultimate criminal? Detection of hybridized DNA fragments by autoradiography. Now more you will understand by looking at this diagram. Look here, students, understand the diagram. That's all is your DNA. I don't know how many of you this is visible. I think it's too light. Students, is it visible? Wait, I'm checking in my phone also. Yes. I also write the full form. Variable number of tandem repeats. Tandem repeats I taught you people. So variable number of tandem repeats. This comes under your satellite, mini satellite, micro satellite is there. That is two types of satellite DNA. One is mini, one is micro. Mini comes between 1 to 60 base pairs. Micro comes 1 to 6 base pairs. Okay. So whatever comes under micro, we say SNSTR, and whatever comes under mini, we say VNTR. Slide blurring is there. So I request all of you to open. Okay, open the NCERT textbook and keep. Yes, correct. Variable number of tandem repeats. That is your VNTR probe. So students, this variable number of tandem repeats, like I told you, I know that no matter what, Praveen will have only 10 tandem repeats, correct? With that also, I can use my VNTR probe to understand, correct? Variable tandem repeats. I'll understand that Praveen has only 10 tandem repeats, no matter if I'm taking his hair, blood or saliva. In a crime scene, I have got 33, or I have got, out of all of it, I have got 25. I can correctly focus that Shreyas was there in the crime scene, correct? Agreed, all of you? So that kind of a method is used. Otherwise, look here students, see. In this, I request all of you to keep your NCERT open. When I give you the notes, this picture will be clear. Students, we have your individual A and individual B. Now, these two people were there in the crime scene. So, we got some blood sample. Luckily, we got blood sample of two people. One was criminal A. We got blood sample of A, and we got some blood sample of B. Okay, and we got one blood sample from the crime scene, which we labeled it as C. We don't know who is this A and B. We have no idea. We went to the history. We understood that this person has some anger with this person. Some jealousy is there with this person. So maybe these two would have fought. Right? These two would have fought, because of which. Okay, did you all like the session? Did you all like the session? No more class. No more class if you people haven't put your attendance. Put attendance quickly. Yes, good job. Okay, students, so we got a blood sample from crime C. That means we got a blood sample from the crime, and that we labeled as C. Now look at individual A and individual B. Okay, look here in your paternal. All of you, eyes on your textbook. Only you should listen to my voice. Okay, eyes on your textbook and try to find out. Now from paternal and maternal. Paternal side there is 3. Okay, numbers I'm writing 3, 4. Okay. These are the repeats, right? We are getting. Here it's around 11. I think this is 7. Okay. Next. Now if you see, this individual A has got from paternal side 3, maternal side 4. Okay, repeats are there, or that differences are there. Now we know that paternal and maternal will not be same, right? Will not be same. It will be different for each of them. Okay, each of them, because our parents are also different. Now look at the other situation, students. In the second one there is 8. Okay, next 4, 3, 7. Okay, again here 8, 7, 4 plus, yeah, I'm doing maths, 5, 11, 5, 2. Okay, these are the numbers we have got. Okay, these are the sequence numbers we have got. Okay, we got 3, 4, 11, 7 and all that. Now what is expected? I am expecting that I do not care about this the ones which are marked in yellow and blue color. I am only bothered about paternal and maternal. Definitely paternal and maternal for this individual A and individual B is going to be different, right? That is going to be my 0.1% could be that difference is because of that. Correct? Our parents are different, that's why our genetics also are different. So from the crime scene, right? From the crime scene, I got a blood sample. I run that with agarose gel. I isolated the DNA. I cut them with restriction endonuclease. After that, I run that on electrophoresis gel. Now, after electrophoresis gel, I have also separated and amplified them, and I've kept it on the southern blotting plate. Okay, now what am I going to see? Now I'm going to observe. If I run A and B on an agarose gel, and if I get fragments on that, I have to compare it with C. Okay, look here. According to my C, according to what blood I got from the crime, I am getting 11, 7, 18,752. Looking at this, okay, students, looking at this, can any of you tell me, one second, yes, done. Yeah, I'm just kicking out our beautiful spammers so it does not disturb you people. Yes, no disturbance, right? What is happening? Yes, okay, done. Yeah, done. Students, so from the crime scene, right? From the crime scene, we understood these are the numbers we are getting: 11,78752, and this is matching. This is matching with the individual B. Now you tell me who did the crime? Who did the crime? Yes, got it? One second. I have a doubt. No, paternal and maternal cannot be same, Jyoti. It's very, no, it will not be. Even if it is, see, when it comes to, marrying from inside the family, there are chances that a lot of genetical issues can come to you. Like, some kind of mutation, some kind of things can come to you. But other than that, you can definitely differentiate. Students, so from this, right, so look here, number of short tandem repeats, that means your STR with the help of your STR number, that means depending on how many repeats can happen. Now this person, individual A, will have three maternal repeats, 4 paternal repeats. This will not be same for any other person. Only this individual A will have this 3 repeats. Can it be same for me? No. The same sequence might not be repeated in me. Maybe I will have 3 repetitions, but the sequence will be different. Correct? One or another difference will be there. Okay? So, depending or looking at the tandem repeats, I can identify easily who was there in the crime scene and who is the actual criminal. Now, who is the criminal? B is the criminal. Okay? Because the blood sample that I got from the crime scene matches with my, look here, matches with the electrophoresis result. Clear to all of you? Inbreeding depression. Yes, inbreeding depression will come. Right? Married from inside the family and all that. Yes, B is the killer. Crime scene solved. Okay? Crime scene solved. One second. Okay, students, imagine I write here one crime scene. Okay, so say there is individual R, Y, okay, and I got saliva samples. Which is, I'll write it here, saliva sample, I will label it as X. Okay, this is the crime scene. You people have to investigate. Now I'm going to write some tandem repeats. I'm just going to write the numbers. Okay, I'm not going to draw, I'll write the numbers. So R has tandem repeats of, from maternal side he has 5, from paternal side he has 7, from chromosome A, B, C something it has 2, he has 3, he has 19, he has 9. Okay, this is the chromosome number. Of individual A. Okay, now let's go to Y. I'll write it in a different color only. Okay, now from maternal side he has got 7, another person got 11, 3, 2, 19, 9. Okay, 19, 9. This is what is the second person. Now in the crime, okay, in the crime scene, I've got a saliva mixed with blood. I'm getting a value. 57. Now tell me who is the criminal. Now you people tell me who is the criminal. Very easy. Very easy. Correct? Very easy to find the criminal. Quickly let me know. Student, this is what I've got from the crime scene. Okay? This is the crime scene blood sample or saliva sample. Okay, crime scene saliva sample. Tell me, is it R or Y? Who did the crime? Yes, very good. Very good. So we understood easily that the crime is committed by this person. He is the criminal. He is the criminal. But students, imagine there is fingerprint of this person. While doing an investigation, we found fingerprints of this person. We found fingerprints of this person. Okay, and this saliva sample I got from the hall. Okay, sorry, I got from the hall. When I'm moving further, I am seeing that in the room, I am getting a slight, very less, very small drops of blood, which has this, which is giving my electrophoresis value as this. So what could you conclude? What could you conclude? Spammer bias, I love his dedication. Yes, kicked out. Done. Done, out. Yes, out, no? Teaching and this, it's too difficult to do, students. Okay, students, tell me, what can you conclude from this crime scene? In the hall, I happen to get this blood sample. I got this as my electrophoresis value. That means electrophoresis bands. Okay, but I'm seeing that in the living room, I'm getting this blood sample. I got one drop of this blood sample. 7-11-3-2. What to do then? What do you understand from the crime scene? Tell me, let me see how much of your CID buddhi is working. All spammers are out. Is there any spammer which I did not notice? Okay. Yes. Students, what is the crime scene? That means both of them are a part of the crime scene. This person's saliva happened to fall in the hall, because of which we got this from the crime scene. But later investigation, we saw blood stain in the living room which showed us this. But still we know that it is among R or Y, that one of them are the criminals or both of them are involved, and they have manipulated the police to make sure that only R becomes the criminal, but later they found out Y also is the criminal. Correct? Both can be. Right? It is a group killing, or they have planned in such a way that this can happen. There are a lot of situations where we do not get blood sample, no saliva, all that is there. That is when we look into the CCTV footage, we look into the phone calls, we look into the text messages that these people do, and all these spammers also, they don't understand that there is cyber crime, there is all of this where people, when if they cross limits, things can go all the ways, right? So in those situations is when we look into the CCTV footages and other things. Otherwise, generally, if there is blood sample, a big murder has happened, and things are going in such a way that like there's other DNA tests that can be done, we definitely use this method. Clear to all of you? Clear to all of you? Yes or no? we have finally finished our DNA fingerprinting. So this is how they understood, because of the presence of VNTR and your STRs. Okay, STRs. Students, just for your knowledge, I'll tell you, satellite DNA can be differentiated. See, satellite, I'm writing it here. See, satellite DNA can be differentiated into mini and micro. Mini and micro satellite. Okay, mini and micro satellite. Now this mini satellite is under where VNTR will come. Okay, micro satellite is where your STRs will come. And with short tandem repeats, here it is variable number of tandem repeats. This variable number of tandem repeats, we use it as a probe. Probe means which will act complementary to this tandem repeats, we use them. Okay, here it comes around 1 to 60 base pairs. Okay, it can, this VNTR probes can be around between 11 to 60 base pairs they will have. This one will have 1 to 6 base pairs. Okay, this is the very small differentiation I wanted to give. Mini and micro, this is actually present in one chromosome. So one chromosome like this will have one portion which is mini, one portion which is micro. And now when we talk about this, we also should know there are GC rich regions and AT rich regions. Okay, GC rich regions are 60%. This I have told you while teaching. I think this chapter only I have told you. GC rich regions and AT rich regions. GC rich region, that is your guanine cytosine rich region, is around 60% of your chromosome. Okay, AT rich region is only 40%. 40%. Okay, so micro will come as GC, mini will come as AT. Okay. Yeah, they will come, this repetitive units or tandem repeats are all coming under non-coding. They are not going to code for protein. Okay, they are all coming under non-coding. They generally, this whole repetitive sequence itself is in non-coding. Okay, this is just for your information to understand, because once I have seen a question about GC and AT rich region. So that is why for you to keep in knowledge, and when you read your NCERT, if you find any doubts, you can let me know. Okay, students? Yes. So we are done with this class. Thank you so much for joining. Quickly like the session and let me know in the comments which chapter you want next. I'm going to take human health and diseases on 7th, which is a Sunday. So I am expecting maximum number of students to come because it's a Sunday and I have kept it at 4 o'clock. So you can take a nap, you can have your breakfast with your family, lunch with your family, and then join at 4 o'clock. Okay. So thank you so much. Okay. Thank you so much for joining, and we will see you all soon. Yes. Yes, I will not be replying in the group as of now. Okay. So I will do it later. Let me take some time. Okay. See, so many spammers are coming. There also they'll come. Why to, spoil our New Year's by such people, right? Yes, thank you so much. Bye. Yes, thank you so much, students. Okay. Yes, okay. So see you all in next class. Do not forget to join 7th Jan. Okay, 7th Jan. I want to see you all for human health and diseases. And if you people are facing, do not worry, your mock test 2 results also will be out. We will tabularize it, and like how we gave you last time, we will do it this time also. So do not worry, as soon as we get the result, we will share it on our telegrams as well as on Vedantu NEET English telegram. So it's just a matter of some time. Give it some time. We will definitely give you the results. And other than that, VDBPs are happening every day. So now also link will be there for molecular basis of inheritance. You can go and solve. Chikungunya and dengue also, I have framed some questions because there's no questions out there. I have put some questions and the team has also added some questions. So you can go solve that also. Okay. This chapter is entirely completed. Molecular basis of inheritance. First part is there, which is around 5 hours. You can see it in 1.5x, 2x if you already know the chapter. This is the last part of this chapter and we are done with this chapter. Entire human health and diseases, I will try to complete it on Sunday. Okay. Yes, thank you so much. See you all. Take care. Bye. Happy New Year to you and wish your family from my side as well and from the entire Vedantu NEET English channel. We also have Diksha ma'am's class at 8 o'clock, so do not forget to join. That's the reason why I have kept my class very short, so we do not drain you people. We always care about your health as well. So if you haven't watched our shorts, also go watch our short. Let us know in the comments how you're feeling it, and we will see you all. Thank you so much, students. Once again, Happy New Year.